In this video, we'll cover how to fill the most commonly used bottles and vials during sample collection. If you have any specific questions about your sampling, contact your project manager. If your kit includes sample bottles for volatile testing, you may find a trip blank or travel blank. These are used to measure any potential contamination that may be present throughout the trip. If your kit contains travel blanks, leave them unopened in your cooler and send them back with the rest of your samples. If your kit includes bottles for PFAS testing, EPA 533 or 5371, you may find a field reagent blank. The field reagent blank detects any contamination due to sampling and environmental conditions. When included, you will receive a bottle of reagent water, an empty bottle with preservatives, marked as a field reagent blank, and bottles for the sample site. If your kit contains a field reagent blank, pour the lab reagent water into the empty field reagent blank bottle near the site of your sample tap. Afterwards, fill your other field reagent blank sampling bottles. For standard plastic and glass bottles, simply fill your container up to the bottom of the neck. While doing so, be careful not to rinse out any preservatives that may be contained in the bottle. For standard glass vials, fill until 3/4 full. Then, use the vial's cap to slowly pour additional sample water into each vial all the way to the top until it forms a dome. For bottles with a 2-stage preservation, there will be a small vial attached to the sampling bottle filled with an acid. When sampling, fill your container 3/4 full with the sample water. Then, add the full contents of the vial into the container. Afterwards, cap your bottle and mix by inversion. Then, continue filling the bottle with the sample up to the bottom of the neck. For vials with a 2-stage preservation, there will be a small dropper attached to your vials filled with an acid. When sampling, fill your vials 3/4 full with the sample water. For chlorinated samples, let your sample sit for 1 minute. Then, add 10 drops of the dropper acid into each vial. Then, using the vial's cap, slowly pour additional sample water into each vial all the way to the top until it forms a dome. Finally, cap the vials and invert to mix. Ensure that there are no air bubbles present in your vial. If air bubbles are present, carefully open your vial and add more sample. For sampling that requires field filtration, a sterile syringe and filter will be provided with a sterile sample bottle. When ready, fill your spare bottle with water from your tap. Next, fill your syringe with sample water before attaching the filter to the syringe and pushing through into the sample container. Repeat this process until the sample bottle is half full. Kits for California may not include sterile syringes. In this case, fill your bottle to the halfway point. For microbiological testing, you may receive one or more 100 milliliter sterile, clear plastic bottles. Right before sampling, disinfect the inside and outside of your tap with alcohol and allow it to evaporate before sampling. While sampling, avoid contamination by making sure the cap and the mouth of the bottle do not come into contact with anything other than the sample water. These bottles should be filled with sample water between the 100 milliliter line and the shoulder of the bottle to accommodate the minimum 102 milliliters needed, with a minimum ½ inch of headspace. Once filled, place these bottles into a separate bag before placing into your cooler.